Okay, so welcome back to this next video in which we are discussing endothelium-dependent vasodilatation. Okay, so we're currently in the process of discussing mechanisms of endothelium-dependent vasodilatation, and we've seen nitric oxide is one of the key molecules that endothelial cells produce, which causes relaxation of vascular smooth muscle cells and hence dilatation of the arteriole. Okay, we've also seen prostacyclin as another molecule which endothelial cells produce and which is sent to the vascular smooth muscle cells to cause them to relax. Okay, now what we want to discuss is a phenomenon known as endothelium-dependent hyperpolarization, or EDH for short. So this is endothelium-dependent hyperpolarization. Now, what I should say is that this is quite a controversial phenomenon. Okay, loads of people have very different ideas about the mechanisms which cause endothelium-dependent hyperpolarization. Okay, so what I'm firstly going to do is just describe what it is, because that at least everyone can agree on. Okay, and then we'll look at the two major prevailing mechanisms, okay, by which endothelium-dependent hyperpolarization probably occurs. Okay, so this is the phenomenon that endothelial cells, which have got these calcium signals occurring in them due to shear stress or potentially due to the inflammatory mediators histamine or bradykinin acting on G protein coupled receptors coupled to GQ, these are able to hyperpolarize vascular smooth muscle cells in some way. They're able to decrease the electrical potential difference across the cell membranes, i.e. make it more negative, okay, and therefore reduce the activation of voltage-gated calcium channels in the vascular smooth muscle cell membranes, and therefore reduce the amount of calcium coming into the cytoplasm of the vascular smooth muscle cells, and thereby reduce their contraction. And this is another powerful way in which endothelial cells cause the vascular smooth muscle cells to relax. Okay, so that's the phenomenon, okay? How it occurs is a complete and utter mess, okay, with loads of different papers out there all claiming different mechanisms for how this works. What I am going to tell you is the main two mechanisms which are your best bets for how this works. Okay, so the first mechanism that I'm going to outline is what's known as the contact-mediated mechanism, or the contact-mediated pathway. Okay, and this pathway only works for the endothelial cells of the arterioles that are actually near to the vascular smooth muscle cells. Okay, so this is the difference. This is the one that's different from all the others. This is the one where the endothelial cells actually do need to be close to the vascular smooth muscle cells, because they actually do need to have contact with them. Okay, they need to specifically form gap junctions with them. Okay, so it's only the vascular smooth muscle cells that are actually very near to an endothelial cell in the arterioles that can actually experience this contact-mediated pathway. Okay, so let me outline this contact-mediated pathway then. Okay, so we'll need two cells now. We'll need our endothelial cell here. Okay, and we'll now also need our smooth muscle cell beneath it. Okay, and now they're going to be contacting each other through gap junctions, which I'll draw in a moment, so I'll just put the basement membrane here. But they're going to have a gap junction despite the basement membrane being in the way. Okay, right. So, basically we start off with the calcium signal inside the endothelial cells here. So calcium has gone up. Now what's that going to do to the endothelial cell? Well, we've discussed many things that does to the endothelial cell, but one of the things that it does is it activates the opening of certain potassium channels. Okay, so there are a family of very special potassium channels which are known as the calcium-activated potassium channels. Okay, now these are tremendously important in the brain. They're important also in immunology, but they're also important in these endothelial cells in the phenomenon of contact-mediated pathway for endothelium-dependent hyperpolarization. So they're the calcium-activated potassium channels. Okay, now There are many different types of calcium-activated potassium channels, but the ones that are believed to be very important in endothelium-dependent hyperpolarization are the intermediate conductance calcium-activated potassium channel, which is written IKCA. Okay, so this will be called the intermediate, this is the I, okay, and then it's intermediate conductance, and then it's just potassium, sorry, not, I get always confused with this, calcium-activated potassium channels. If I ever say potassium-activated calcium channels, just reverse it round, okay? It's calcium-activated potassium channels, but I'm bound to say that at some point. I managed to stop myself from saying it just then, but I might miss it later on. So this is the intermediate conductance calcium-activated potassium channel, okay? Now, the other one that's important in this is this small conductance, the calcium-activated potassium channel. So this is the small conductance calcium-activated potassium channel, the SKCA. Okay, right. Now, let me describe the structure of these to you, okay, because each of these channels is actually a tetramer of 4 separate proteins. Okay, so let me just draw one of these a little bit out in more detail. Okay, so let's say this is the cell membrane here, and here is our calcium-activated potassium channel, which is either going to be an intermediate conductance calcium-activated potassium channel or a small conductance calcium-activated potassium channel. And basically they're not just a single protein, instead they're actually 4 proteins all bound together. So each one of these channels, the IKCA or the SKCA, is actually a tetramer. Okay, right, so these 4 subunits that make up the tetramer in the IKCA channel, those subunits are KCA 3.1 subunits. So there are loads of different subunits that you can use to build calcium-activated potassium channels. If you're going to build an intermediate conductance calcium-activated potassium channel, you need to use 4 of these KCA 3.1 subunits. So each one of these subunits here that makes up an IKCA channel needs to be KCA 3.1 proteins. Okay, meanwhile, for small conductance calcium-activated potassium channels, there are actually 3 different subunits that you can use to build them, okay, which are known as KCA 2.1, KCA 2.2, and KCA 2.3. However, the ones that are important in endothelial cells seem to be the ones that are constructed out of 4 KCA 2.3 subunits. Okay, so in the endothelial cells then, there are these calcium-activated potassium channels, which are either intermediate conductance calcium-activated potassium channels built out of 3 KCA 3.1 subunits, and you don't have any choice in that. Intermediate conductance calcium-activated potassium channels always are built out of 4 KCA 3.1 subunits. Okay? Small conductance calcium-activated potassium channels, there are 3 possible subunits you can use to build those. The ones that are important in this phenomenon seem to be the KCA 2.3 channels. Okay, right, so let's represent both these types of channels here. Okay, so here they are. This is our calcium-activated potassium channel, which is either the intermediate conductance calcium-activated potassium channel or a small conductance calcium-activated potassium channel. Okay, now the key thing here is that they are calcium activated. So when the calcium signal occurs here, it causes these things to open, okay, whereas they were closed before. And now they're going to allow potassium ions to move across the cell membrane. Now, which way does potassium move when you open a potassium channel? Well, it's usually out of the cell, and indeed it is out of the cell in this case. So potassium is going to move out of the cell, and that's going to hyperpolarize the electrical potential difference across the cell membrane of the endothelial cell. And you might be saying, well, hang on, I thought we were going to hyperpolarize the electrical potential difference across the vascular smooth muscle cell. We are, but that's coming in a moment. Okay, so we're first going to hyperpolarize the electrical potential difference across the cell membrane. So when you move potassium out, you're moving positive charge out of the cytoplasm. So going back to our electrical potential notation, you are effectively reducing the electrical potential intracellularly because you're moving positive charge out, and you're raising the electrical potential extracellularly, okay, the E for electrical potential, O for outside, okay, and effectively what is that doing? Well, if we go back to this diagram, remember, here is the electrical potential outside, here is the electrical potential inside. You usually have to step down, okay, but now we're raising this one up, we're reducing this one down, so the amount you're going to have to step down is going to go up. So we're going to hyperpolarize the electrical potential difference across the cell membrane. Okay, now, why is that going to cause a hyperpolarization in the vascular smooth muscle cell? Well, basically, there are electrical connections between the endothelial cell here and the smooth muscle cell below. Okay, and I'll draw this like so. So this is what's known as a gap junction. Okay, and they are effectively electrical windows between the endothelial cells and the vascular smooth muscle cells, and they obviously exist in other places in the body as well, importantly in the brain as electrical synapses. Now what are they? They're effectively an ion channel through which ions can move from the cytoplasm of the vascular smooth muscle cell into the cytoplasm of the endothelial cell, or vice versa. Okay, right. So this is a gap junction. Okay, so what does this mean? Well, this means that when you change the electrical potential difference across the cell membrane of the endothelial cell, it's going to cause a change in the electrical potential difference across the cell membrane of the vascular smooth muscle cell as well, okay, because they are electrically connected in this way. Now, these gap junctions have a special name. They are called Mayo endothelial gap junctions, or MEG-J, like so. So this stands for Mayo endothelial gap junctions. Okay, so let's think about what might happen when we have this myoendothelial gap junction. So what have we just done? We have now decreased the electrical potential intracellularly inside the endothelial cell. Okay, that will now mean that the electrical potential intracellularly is no longer in equilibrium with the electrical potential in the vascular smooth muscle cell, because we've moved this down. Okay, let's imagine that they were previously in equilibrium, since they're connected electrically like this. Okay, but now once the electrical potential is lowered in the cytoplasm of the endothelial cell, it's now going to mean that positive charge is going to want to move from the cytoplasm of the smooth muscle cell here into the intracellular compartment of the endothelial cell. So this might be potassium ions that might want to move from here to here, and that of course is going to reduce the electrical potential inside the vascular smooth muscle cell, and that's then going to lead to the hyperpolarization of the electrical potential difference across the cell membrane of the vascular smooth muscle cell. Okay, right. So these myoendothelial gap junctions can transfer the hyperpolarization across the membrane of the endothelial cells to the vascular smooth muscle cells that are connected to the endothelial cells in this way, but not to the ones that are further out. Okay, so in vascular smooth muscle, the smooth muscle cells are not all electrically connected, unlike in places like the bladder, where all the smooth muscle cells are connected by gap junctions to one another to form a massive great syncytium. In blood vessels, it's not the case, okay? So it's not the case that if this one here hyperpolarizes because it's in contact with the endothelium, that vascular smooth muscle cells on the outside, like this one here, will also hyperpolarize. So it's only those vascular smooth muscle cells that are actually in contact with the endothelial cells in the arterioles that can actually experience this contact-mediated pathway of endothelium-dependent hyperpolarization. Okay, right. So that's the contact-mediated pathway of endothelium-dependent hyperpolarization. What we're now going to turn our attention to is looking at another pathway of endothelium-dependent hyperpolarization, which involves a diffusible molecule. Okay, but I think we'll call it there for this video, and we'll look at that in the next video.